So till last video we were done with another, like before that we were done with the experiment, the Griff's experiment. Today we are going with the Hershey chess experiment. Okay, so let's check it out. What we'll see, first thing, this experiment was done by two scientists. This Hershey word, this means this was, if we talk about his complete name, he was Alfred Chase. And when we talk about Chase, this one, that was Martha Chase. Martha Chase, which was simply a girl scientist, and this one was a boy scientist, right? Okay, so let's start. First thing, this experiment was conducted in which year? It was conducted in 1952. Then he took bacteriophage for his experiment to identify DNA as a genetic material. Bacteriophage. Why he took bacteriophage? Because this is quite similar, because its composition, which is made up of DNA plus protein. So, like chromosome, this is also made up of DNA plus protein. That's why he took this one. Bacteriophage is simply kind of a virus. So, if I just make any structure, this is the structure. This is the structure basically. Right, I hope it will be clear. Then let's label it out. First thing, you will see this a protein capsid. This outer one called as a capsid. Inside this we have a DNA, hereditary material, but at the time no one was known. This is T2 bacteriophage. T2 bacteriophage. So we have chosen virus. Why we've chosen virus now? Because simply virus is the only organism which is simply, which cannot produce, which cannot respire, which does not have DNA, and it only have a DNA, except that it does not do any process if it's outside the host body, and when it comes in the host body it become alive and like do all the functions. Otherwise virus is kind of good person. I mean, it does not do anything, it only have a DNA, but when it goes in the host body it do so many functions, which we'll see. So if I just enlarge this DNA, what you will see, this basically this is virus simply, this which is going to be inserted here. So this DNA has been inserted. When DNA insert in the body, this means it start copying themselves, like multiplying. DNA copy by the help of DNA polymerase, many copy would be formed, right? Then e coli, which is we use the E. coli to produce DNA here. At the time DNA was not known what hereditary material is this, because they were searching to find out is that a hereditary material. So this, till this one, they took a virus and inserted, so it started making itself copies and so many processes. But at that time DNA was unknown, so till here it was either DNA or RNA or a protein, no one was known. So how they came to know this? Then the experiment started. What was his experiment? Basically, he took two jars. He took protein and sulfur, and that he realized when we were taking kind of radioactive DNA. This is radioactive DNA, radioactive DNA, and second one. In this one, it have taken phosphorus, radioactive phosphorus, and here we are going with another one. In this one, radioactive sulfur, radioactive DNA, which is sulfur, it was taken. So sulfur basically is kind of same like protein which have amino acid and same formation like that. So what we observed, when we added phosphorus, what was the result? Let's check it out. When we added phosphorus, what we saw, that if this is the radioactive DNA, here comes the radioactive DNA. And same like here, when we added, so what we observed, here non radioactive DNA was found. This means the copy of DNA was only working on phosphorus, not on the sulfur. This means all other new phages would be formed by the phosphorus, not the sulfur. Till here it was clear. So what they do, they started an experiment. I can show you here. Okay. First one, here is the phage, label phage. Here first one, radioactive protein was taken, inserted. Then we did centrifuge process. Centrifuge is a process where we mix all the things, and like protein becomes separated when we mix together. And the second step was agitation or something. uh, Phages were unknown. Here also third one, right? So the second step was radioactive phosphorus. And yes, before I just tell you that radioactive phosphorus and sulfur 23 was taken. So here radioactive DNA, centrifuge process, and what they observed, after centrifuge there was a radioactive phage where DNA was forged, or we did pellet. When we saw this pellet, it was simply kind of white in color, it was red in color, not red but the pink in color. So if I make something more over here, after this one, the centrifuge process, what we observe, here's the centrifuge process. What observed here was the kind of structure which was, they were radioactive, and on the upper side here non-radioactive protein, like non-radioactive part was there, non-radioactive, right? And when comes here, what was there, here was on the upper side, here it was opposite. Here radioactive was on the upper side, radioactive, and when comes this downward part, non-radioactive part, non-radioactive part. So this was simply difference between this one. So let me add picture of this one again, then we'll read it out. So let's read it out. What's written? First one, there were these Alfred Hertz and Martha Chase. These both scientists did experiment in 1952. Experiment was done on a bacteriophage. Why? Because bacteriophage composition and chromosome bacteria are quite similar. They are both made up of DNA or protein. So during lactic cycle, the division phage, what we observe, which of the molecule phage work as a heredity material. So Hershey and Chase developed experiment finding out heredity material of bacteriophage. So what was taken, the experiment, there was a DNA in the bacteriophage with P32, phosphorus 32, and sulfur 23. If I'm not, sulfur is 35. Then what we observed, the radioactive, these were done, viruses were promoted. The new phage which was coming out from this one, from host, that was P32, not S35. So what they observed, that S35 was founded on a medium. This means result, it was clear that the P32 containing DNA enter in a host body, and rest, this is also as a heredity material. Till here I hope it will be very much clear. This was said by Hershey Chase experiment. Thank you for watching. Bye bye.